Hey guys, welcome back to the second part of Unit 1, Lesson 1's online virtual lecture here. So in this segment, we're going to be learning about the way biologists do their work. And that's called the scientific method of inquiry. And it's beautiful. I live by it. To me, it's just the greatest way to figure out information. But it's very humble because, okay, ponder this. Why do we do science? We do science so we can learn the truth about things, right? Except the scientific method of inquiry says we will never know the whole truth about anything because there could always be other explanations we haven't thought of. And so we go out and we do more work, and it's a circular process. It's awesome. I love the scientific method. I've lived by it since I was a little kid. Well, we're going to introduce you to the steps of the scientific method, and there's no official way that the scientific method works, but there's the most common way, and that's the one I'm going to introduce you to. But depending on what field of science or field of biology you're in, things could be slightly different. But for the most part, we can say it goes with observations, questions, hypotheses, experimentation with the results, conclusions, theories, and then back around, starting with observations again. Okay, how does this work? Let's take an example. Okay, let's say you have a pet dog, and his name is Pluto. Hey, Pluto. And you see that he's scratching himself, and he's going crazy. And you're like, okay, so he's scratching himself, going crazy. Well, you just watched that, so you actually did the first step of the scientific method, and that was to make an observation. Observation, dog scratching. Well, we humans are pretty curious creatures, and we're not satisfied with just making observations. We like to ask questions about what it is that we observed. So that's the second step of the scientific method. So we ask a question about our observations. Maybe it's a what question, maybe it's a how question. In the case of Pluto the scratching dog, what do you think kind of question is that? Well, I'd want to know why. Why is he scratching? So you say, I keep a clean house, so why is he scratching? So I've done the second step of the scientific method. Well, again, we curious human creatures are not very satisfied with just asking the questions. We want the answers. So we want to come up with an answer to the question we just asked. And if that answer is a guess, but it's an educated guess, meaning takes into consideration some knowledge we had somewhere in the past, then in science we call that a hypothesis, or plural is hypotheses. So we made an observation, we asked a question of why, and now we're going to come up with an educated guess, a hypothesis, to answer that question. Now, what kind of hypothesis could you give that would be an answer as to why he is scratching? Well, some of you out there, I could hear you, said maybe he has fleas. Great observation. In fact, I'm going to grab my little handy-dandy PowerPoint pen here, and I'm actually going to, well, hopefully it'll work. Let's see. I'm going to write fleas. Wow, it's really hard to write with a trackball here. Okay, I'm going to write fleas. So maybe he has fleas, and that's why he's scratching. Now, is that the only possible hypothesis we can come up with that would explain why he's scratching? No, right? What would be some others? Come out with some. Shout them out. Can't hear you. Shout it out louder. Maybe he has dry skin. Maybe he has a behavioral tic. You know, he's just like this. Maybe he's starved for attention. He's just trying to get your attention. Maybe it's not fleas. Maybe it's lice. Who knows what the reason is why he's scratching? So there's actually an infinite amount of explanations for that observed phenomenon. There's an infinite amount of hypotheses, and we can never test them all. One thing, we don't have time, we don't have money, etc. So we're going to just tackle one of those hypotheses for now. Let's just make it fleas, keeping in mind that there could be others. So an educated guess, maybe he has fleas. Again, there could be maybe he has dry skin or whatever, but we're going to just tackle fleas for right now. Okay, so here's the question. How could we determine if our hypothesis that fleas are the cause of the scratching is valid? Now, here's the beauty of the scientific method. You can never prove anything true in science. You can never prove anything true. There could always be another explanation you hadn't thought of. All we can do is get closer to the truth. So, in other words, we can either support our hypothesis with the evidence, or we reject that hypothesis if the evidence is contrary to it. So I think you'll see what I mean in a minute. Well, how are you going to determine if you've supported or rejected your hypothesis? Well, you're going to do the next step of the scientific method, and that is to do an experiment, to come up with some experimentation. And how could you do that? Well, there's a lot of ways you could do an experiment, but there's two things that every good experiment should have: control and replication. Say that with me: control and replication. So, tackling our hypothesis of the fleas, what is an experiment we could do that would show not just correlation, but causation, that fleas are the cause of the scratching? Well, how many of you out there said, give Pluto a flea collar? So if flea collars are supposed to kill fleas, so put the flea collar on Pluto and see if he stops scratching. Okay, so let's do that. Here's Pluto. Okay, it's actually a zebra, but we'll pretend it's a dog. Okay, so we're going to go and stick a collar on Pluto. I haven't actually tried this before. Hopefully it'll work. Okay, so we're sticking this flea collar on poor Pluto, and lo and behold, he stopped scratching. We've proven that fleas were the cause of the scratching, right? No! We haven't ruled out other hypotheses. That was just a correlation. It wasn't causation, because how do we know that the real reason why he's scratching is because he's starved for attention? And so by putting that flea collar on Pluto, I've actually given him attention, even if it's bad attention, and therefore he's happy, he stopped scratching. We don't know. All we've seen is correlation. You put the flea collar on, he stops scratching. So what we need to do, and I didn't tell you that we could actually do this, so in my world we can, my magic virtual world here. We're going to wave our magic wand, and we're going to suddenly clone Pluto. Now, I only have one of these, so just pretend he's cloned. So now we have a Pluto with the flea collar and a Pluto without the flea collar. And we want to make sure these guys are exactly the same. So that means they look the same, they were born the same, they born at the same time, so they're the same age, they act the same. They're basically clones of each other because we want to isolate the variable of interest. And in this case, the variable of interest is whether or not he gets a flea collar. And so that is called control, when we're controlling for all the variables except the one of interest. Now, the problem is we stuck a flea collar on our treatment Pluto, but we didn't stick a flea collar on our clone Pluto. And what's wrong with that? Well, how do you know that there's not lotiony properties in this flea collar that soothe his dry skin? And the guy that didn't get that collar didn't get that lotion, so he's still scratching, but it's because of dry skin, not because of fleas. So in other words, we haven't isolated that fleas are the cause of the scratching. So what we want to do is take our control Pluto and treat him exactly like the treatment Pluto, except leaving out the variable of interest. So how we're going to do that is we're going to put an identical flea collar on the control Pluto, except it won't have the flea medication. So he's exactly like this guy was, but you know, he's getting the same amount of attention, so we're controlling for that variable. It would have the lotiony properties that this one would, so we're controlling for that variable. So we're treating them exactly the same, except leaving out the variable of interest, which now is the medication, the flea medication on the collar, not the collar itself. Sounds good, right? Okay, so we've gone to the next step. You know, we bought a flea collar, put it on Pluto, but now we need to do some more cloning. So we have a control and we have a treatment Pluto, but we only have one of each. That means our sample size is 1. And what's wrong with that? Well, let me put it this way. Let's say you guys take your first assessment, and I get a call from my dean, and my dean says, Bree, I need to know how your students are doing. They just took their first test. I say, okay, so I'm going to call up student X and say, student X got a 97% on that first assessment. My students are doing great. Well, the dean would probably raise an eyebrow because that student may not be representative of the whole population of students in the class, right? There might be a whiz kid, right? So how are we going to control for that? Well, if this was a class, it would be more accurate. I'd get closer to the truth by taking the scores of everybody in the class, adding them up and dividing by the number of students, right? Taking an average or a mean. Well, so I got to do that in science too. How do I know this Pluto and my control Pluto are representative of all dogs or even all Plutos in the world? How do you know there's not something weird about this one? So we're going to wave my magic cloning wand again, and bling, I'm going to make 30 treatment Plutos and bling, make 30 control Plutos. Now I have 30 in each category, and so I'm going to put their collars on. This guy gets the flea medication, this group doesn't, and see if they stop scratching. And so I'll add up the results of the treatment Plutos, divide by the number of treatment Plutos, which is 30. Same in the control Plutos, add up the results for all the Plutos in the control group and divide by 30. So now I'm going to be comparing averages between the two groups, and that gets me much closer to the cosmic truth out there of what the reason why Plutos scratch really is. So in other words, I have now added replication in there. So I got control through my control Pluto that gets treated exactly like this guy does, except the variable of interest, and I have my replication, 30 Plutos, exactly the same in each group. All right, so I look at my results, and lo and behold, I see that none of the treatment Plutos are scratching, all the control Plutos are still scratching, and my carpet is a flea graveyard. Have I proven that fleas were the cause of the scratching? Well, I've drawn conclusions that my results are consistent, i.e. they support my hypothesis that fleas were the cause of the scratching. But I have not proven it true because, why? There could always be another explanation you hadn't thought of before, right? For example, have we ruled out dry skin? Well, they still both get the lotion, right? So we haven't ruled out that dry skin could be the cause of the scratching. But let's say all science labs all over the world studying Plutos set up the experiment the same way, and they all come up with the same conclusion that yes, we have failed to reject the hypothesis that fleas are the cause of the scratching. We haven't proven it true, but we failed to reject. Our results are consistent with that. So now we can create what's called a theory. And this is going to be the theory of the scratching dog, dogs that scratch out fleas. Okay, so we went through all that, collected all that data, and now we're at a theory? What's going on here? Well, a theory in science is very, very different from a theory in other disciplines, such as a court of law in the United States. So if a lawyer in a court of law says, I have a theory that this defendant here is guilty, that's more like a hypothesis in science, right? A theory in science is something that's gone through tons and tons and tons of rigorous scientific protocol using the scientific method, and no scientist has been able to reject the original hypothesis. Therefore, it's good enough to be called a theory. So some of you may have heard of something called the law of gravity, right? Well, there's not really any such thing as a law in science because everything is open to new evidence that might present itself that we haven't even thought of. So, for example, gravity is actually a theory because we can't test it at every single second in every single place of the earth. It is conceivable that somewhere I might drop these keys and instead of falling at 9.8 meters per second squared, they float, or at least they don't fall at 9.8 meters per second squared. Maybe it's a different acceleration rate. So let's test it. Okay, if I drop the keys, oh, they did fall. Okay, so our results there were consistent with the theory of gravity, but we still haven't proven it true because there could be somewhere on the Earth where things don't fall at 9.8 meters per second squared, and we just haven't tested that yet. But that leads us to bring this back around because some lowly graduate student out there is going to say, well, I've observed something that might not be consistent with the theory of gravity or the theory of the scratching dog. And so that person's going to go out and make more observations, ask questions, come up with a hypothesis, develop an experiment, come up with the conclusions, etc. And so the process of science just keeps going and going and going. And that's what's beautiful about it. We get closer and closer and closer to the truth, but we never get there because you can never prove anything true in science. All we can do is support it. So is the scientific method of inquiry the best method of inquiry to find out things? Well, that's where it gets into your own personal ethics and what you think. But it's worked pretty well so far. We've made amazing discoveries, and we've made cures for lots of diseases, vaccines against diseases. So, it certainly seems to work in a sense, but you'll report on the discussion board what you think of the scientific method of inquiry. So the cycle continues. New observations lead to testing old theories, and that's the beauty of science. Okay, so let's get a little bit more into actual experimentation. I gave you a silly example with our Pluto dog there, where we had a magic wand where we could clone him. But let's try to make one a little more realistic. Again, keeping in mind that the purpose of experimentation is to get to causation as close as possible and not just correlation. And some of the best studies involve both lab studies and field studies, depending on what the field of biology is. But again, I mentioned that we had an experimental or a treatment group of Pluto. So these are the treatments where you take the subject, the organism you're working with, Pluto in that case, and you manipulate everything except for the variable of interest. And a control group where you do everything you did to the treatment group except you leave out the variable of interest. And then usually experiments involve some kind of analysis, usually with statistics. So we took averages or means. That's a kind of statistic. Some involve more fancy ones like analysis of variance or who knows what. So let's run an experiment. Let's say we're going to take some corn. And we notice that we're out in the field, tilling the field, and it seems that when we add nitrogen-containing fertilizer to our plants, they grow better. So that's our observation. And so now we really want to know, does in fact adding the nitrogen-containing soil to the plant help it to grow? And let's say our hypothesis is that plants that are given extra nitrogen in the fertilizer grow better than plants not given that extra fertilizer. So how could we set up an experiment? So what I want you guys to do is you can pause this, see if you can think about it, jot down how you would set up an experiment to test the hypothesis, either support it or reject it, that plants given extra nitrogen grow more than plants not given extra nitrogen. Okay, maybe you came up with something like this. You had a treatment group, right, the experimental group, and you had a control group, and you said, okay, I'm going to have a plot of land here and a plot of land here, and I'm going to plant 12 corn plants in the treatment group's plot and 12 corn plants in the control group's plot, okay? I'm going to water each plant every week, so maybe a certain amount, 2 liters of water, or maybe you decide to do it as needed. But then you're going to say each plant's going to get 100 grams of nitrogen-containing fertilizer every week, okay, or maybe the whole plot, or whatever you decide. And in the control group, I'm going to also give them 100 grams of nitrogen every week, just like I did before, 100 grams of fertilizer every week, just like I did the treatment group. But in the control group, I'm going to leave out the nitrogen. So they're treated exactly the same. You trample around the plots exactly the same, except you leave out the extra nitrogen in the control group. Okay, so how many of you came up with something like that? Me. Okay, not bad, right? Okay, you got your control, you got your replication because there's 12 plants in there. Or do you? Do you really have good replication in this experiment? Some of you might say, okay, well, maybe I needed more than 12 plants, whatever. But what actually is your sample size in this case? Some of you might think it's 12 or something else. Well, in actuality, your sample size in this experiment you just set up is one plot. One plot of treatment, one plot of control. So if you were to say that your sample size was 12 instead of, because you had 12 plants instead of the one plot, you'd be doing what's called pseudo-replication. And that's done a lot by mistake, even in peer-reviewed public journal articles. So it's a very bad thing because you're treating something as a representative of a whole, but you're not actually dealing with a large population size like you thought. So here you have one plot. How do you know there's not something weird about that plot? How do you know that plot is representative of other areas of land? So we'd want to replicate the plots. Maybe you could only access enough land to get 3 plots of the treatment group and 3 plots of the control group. Each plot contains 12 plants treated exactly the same way, yada, yada, yada. So you got a sample size now of 3 plots, each with 12 plants. The more the merrier, but that actually wouldn't be a bad sample size given this kind of experimental protocol. Okay, got it? So 2 things important to all experiments, control and replication. Good. Okay, last point to talk about in this first lesson's lecture, and that is the metric system and quantitative analysis. So when you're dealing with things in science, you want to be very precise. The idea is if you're carrying out an experiment and you write it up, you want to write the method you used and how you conducted your results and your analyses in such a way that so someone on the other side of the world speaking a different language and only using a translator could replicate your study. And so we want to make sure things are quantitative measurements. Okay, so if you use words that are relative, like big, small, heavy, light, okay, it leaves in the mind of the reader how big, how small, how heavy, how light. So you want to use a quantitative description that will tell you how much of these descriptive words you're referring to. So those aren't very helpful. Instead, we want to do quantitative descriptions like 7 meters tall. That tells me how tall. 1 meter tall. 100 kilograms, how heavy, right? 1 gram. So these kinds of things where you're going to have both a number and a unit. So that tells you a quantitative description of what you're talking about. And you'll notice that I put things in meters and kilograms and grams. The idea is that we want to keep things in the metric system of measurement. So in this class, we are going to deposit our feet here and pick up our meters here. We're going to use the metric system of measurement in this course because this is the system of measurement that scientists use because it's understood all over the world. In fact, in the United States, we typically use the English system of measurement in everyday life. That would be things like a yard, right? A yard was as long as the King of England of some year's arm, and a foot was the size of his foot, and et cetera, et cetera. It doesn't make any sense because every person's different, right? The metric system allows you to convert via units of 10, and it's very, very, very helpful. So we're going to learn the metric system and how to do some calculations in it. So we're going to keep things clean and metric. So in this course, no cussing. We're going to not use any of these 4-letter words like inch, foot, yard, mile, pint, acre. We're going to keep things clean and metric. Okay, so again, 2 important parts of every metric measurement. The number, which tells the comparison, like 7 versus 10, and the unit to tell the scale, kilogram versus gram. So we as Americans are kind of crazy to use the English system of measurement. Most of the countries of the world use the metric system. All of science uses the metric system. So only America and one other country, I think it's like Micronesia or something, uses the English system of measurement. Even England doesn't use the English system of measurement. So the rest of the world, we use metric. It's based on factors of 10. It's the standard for science, and it's standardized by an agency it does what's called the SI units. So it's an international system of units, and it's located in France, and that's where they standardize what, for example, a gram is, what a meter is. So it's all standardized, and it's easy to calculate these things based on units of 10. And so here is how you do the metric system. So you always start with a base unit. Okay, so for measurements of length, we use the meter, and this is the European spelling. In America, we say M-E-T-E-R. But we use the meter as the base unit for length in the metric system. So a meter is about yay big. It's basically about the height from the floor to a typical doorknob. That's a meter. For measurements of mass, which is kind of like your weight minus the gravity. So for example, your mass and your weight are the same on Earth, but different in outer space where you don't have the effect of gravity. So gram is the base unit for mass. Liter is the base unit for volume. Second is the base unit for time. And in the case of biology, Celsius is the base unit for temperature. Now, there is actually another unit called Kelvin, which the chemists use. We're not going to worry about Kelvin. We are using Celsius to measure temperature in this class. And you'll see that this base unit is stuck in the middle of this chart here. And as you go up from that base unit, things get bigger. So 1 meter is equal to 10 decameters, or 100 hectometers, or 1 meter is equal to 1,000. Going down, so as you go down this chart, things get smaller. So 1 meter is equal to 0.1 decimeters, or 0.01 or 100th of a centimeter, etc. So, and 1,000th of a millimeter. And so here's the abbreviations for these SI units here. This is kind of cool. Look at nano here. Nano is 1/1 billionth, or 0.000000001 meters. So super small. And you know what, nanotechnology now, you may have heard a little bit about it. We'll talk a little bit about it later. You know, that's the level you're dealing, so small. So when you get down into the micro level and the nanometer, you're dealing with highly microscopic things. So bacteria are measured in microns or micrometers. And we use this little Greek symbol here called mu to measure those. So 1 micrometer is 1/1,000,000th of a meter. Itty bitty. So let's take an example of this. Here's a typical ruler. It has both English units and metric units on it. So the big ones are English system. These are inches. Okay, but let's look at the metric system. These smaller things are centimeters. So 1 centimeter is equal to 1/100th or 0.01 meters. In other words, there's 100 centimeters in 1 meter. Centi means 100. So how could you figure that out? Let's say I have 5 centimeters, and I want to figure out how many meters that's equal to. So every time, remember with the metric system, every time you go up or down a unit, you're talking about a factor of 10. So if I go 10 times 10 till I get to meter, 10 times 10 is 100. So there are 100 centimeters in a meter. Or if I go back the other way, from meter to centimeter, 10 times 10, but I'm going down, so I'm going to move the decimal point to the left. So I'm going to go from 1, I'm going to go, imagine I had let's draw this here. Let's say I had a 1 here. Let's say I have 1.0 meters. To figure out how many centimeters that is, since I'm going down, I'm going to move the decimal point to the left. Okay, so that's how you do the metric system, and we'll practice some things like dimensional analysis if you're taking the lab course. Anyway, thanks for joining me for the first lecture of Unit 1, Lesson 1, and now you can go on to Lesson 2 of Unit 1 to learn about the chemistry of life. Take care.